హలో ఎవ్రీవన్, వెల్కమ్ బ్యాక్ టు మై ఛానల్ కాంపిటేటివ్ బాటనీ. ఈ వీడియోలో మీరు సెకండ్ మేజర్ 2 పేపర్ అయినటువంటి ఇంట్రొడక్షన్ టు అప్లైడ్ బయాలజీ నుంచి 9th టాపిక్ ఫిజికల్, కెమికల్ అండ్ బయోలాజికల్ మెథడ్స్ ఆఫ్ జీన్ ట్రాన్స్ఫర్ బయోటెక్నాలజీ యూనిట్ నుంచి ఆబ్జెక్టివ్ టైప్ ఆఫ్ క్వశ్చన్స్ చూడబోతున్నారు. లెట్స్ సీ ద క్వశ్చన్స్ వన్ బై వన్. మల్టిపుల్ చాయిస్ క్వశ్చన్స్. ఫస్ట్ కేటగిరీ, వాట్ ఈస్ ద ఎలక్ట్రోపోరేషన్ ఇన్ ద కంటెస్ట్ ఆఫ్ gene transfer? Options are introduction of genes using electric fields to create pores in the cell membrane, transfer of genes using a needle, introduction of genes using specific enzymes. So the correct answer is first one. So introduction of genes using electric fields to create pores in the cell membrane is the correct answer. Let's see the next question. Which of the following is an example of physical gene transfer method? Options are electroporation, agrobacterium mediated transformation, liposome mediated transfection. So the correct answer is first one. Electroporation is a physical gene transfer method. Let's see the next question. Define biolistic transformation. So options are discovery of genes using electric fields, introduction of genes using a needle, gene transfer using high velocity particles. Yes, the correct answer is gene transfer using high velocity particles is a method of biolistic transformation. Let's see the next question. In microinjection, how are genes introduced into cells? Options are high velocity particles, through the use of electric fields, direct injection of genetic material into the cell. So the correct one is direct injection of genetic material into the cell is the correct one. Let's see the next question. Which technique involves the direct injection of genetic material into a cell using a fine needle? So the correct answer options are electroporation, microinjection, agrobacterium mediated transformation. So the microinjection is nothing but direct injection of genetic material into a cell with a needle. Okay, so let's see the next question. What is the limitation of microinjection as a method of gene transfer? Options are low precision in targeting specific cells, requirement for complex equipment and expertise, high risk of immune responses. So the correct answer is requirement for complex equipment and expertise. సో నీడిల్ ద్వారా లేదా మైక్రో ఇంజెక్షన్ ద్వారా జీన్ ట్రాన్స్ఫర్ చేయాలంటే ఇట్ రిక్వైర్స్ కాంప్లెక్స్ ఎక్విప్మెంట్ అండ్ ఆల్సో ఎక్స్పర్టైజ్ కూడా ఉండాలి. సో లెట్స్ సీ ద నెక్స్ట్ క్వశ్చన్. So which chemical method is commonly used to introduce foreign DNA into bacterial cells by making them more permeable to DNA molecules? The options are liposuction, microinjection, calcium phosphate transfection. So the correct answer is calcium phosphate transfection is the correct answer. Let's see the next question. Identify the chemical method that is commonly used for introducing genes into mammalian cells in culture. So the correct options are electroporation, calcium phosphate transfection, agrobacterium mediated transformation. So the correct answer is calcium phosphate transfection. It is a commonly used method for the introduction of genes into mammalian cells. Okay, let's see the next question. What method of gene transfer involves the use of electric pulses to create temporary pores in the cell membranes for DNA uptake? Options are transformation, transduction, electroporation. So the correct answer is electroporation is the correct answer. Okay, so it's a physical method actually. So what is the role of calcium phosphate in the process of transfection? So options are creating pores in the cell membrane, stabilizing DNA and facilitating its uptake by cells, inhibiting gene expression. So the correct answer is calcium phosphate that stabilizes the DNA and it facilitates its uptake by cells. Okay, so that's why calcium phosphate used. Okay, so let's see next question. Agrobacterium tumefaciens is often used in which type of gene transfer method? Options are electroporation, microinjection, biological gene transfer. Yes, agrobacterium tumefaciens gene transfer method belongs to a biological gene transfer method. Okay, so that is the answer. So let's see the next question. In biological gene transfer, which organism is often utilized as a carrier to deliver foreign genes into plants? As I already told you, Agrobacterium tumefaciens is a bacterium that is generally utilized for the gene transfer. Okay. Let's see the next question. Which gene transfer method is widely used for the genetic modification of plants? Options are electroporation, ballistic transformation, agrobacterium mediated transformation. Okay, so let's see the next question. Generally, widely used technique is agrobacterium mediated gene transfer method is widely used to transfer the genes into higher plants. Okay, what is the primary role of agrobacterium tumefaciens in genetic engineering? So it direct the injection of genes into cells. It incorporates of genes into lipid vesicles. Agrobacterium mediated transformation. So the correct answer is agrobacterium mediated transformation is the correct answer. Let's see the question, next question. Which plant pathogen is commonly used as a vector for introducing genes into plants? Options are tobacco mosaic virus, agrobacterium tumefaciens, herpes simplex virus. So the correct answer is agrobacterium tumefaciens is the correct one. Let's see the next question. What is the natural function of agrobacterium tumefaciens in nature? So it causes diseases in plants, promotes plant growth, transfers genes into plants. So the correct answer is it transfers genes into plants is the correct answer. Okay. Let's see the next question. In agrobacterium mediated transformation, which genetic material is integrated into the host plant genome? Options are plasmid DNA, bacterial chromosomes, host plant mitochondrial DNA. So the correct answer is plasmid DNA is the correct one. Let's see the next question. Which of the following statements about agrobacterium mediated transformation is true? Options are it is limited to dicotyledonous plants. It cannot be used for the genetic modification of plants. It is effective for both dicots and monocotyledon plants. So the correct answer is it is effective for both monocotyledonous and dicotyledonous plants. Okay. So that is the correct one. Let's see the next question. What is the potential disadvantage of using viral vectors for gene delivery in gene therapy? Options are high efficiency in delivering genes, risk of immune responses, integration issues, compatibility with all cell types. So the correct answer is risk of immune response and integration issues is the correct option. Let's see the next question. What is viral transduction in the context of gene transfer? Options are delivery of genes using electric fields, introduction of genes using a needle, incorporation of genes into a viral vector. So the correct option is incorporation of genes into a viral vector is a transduction method. Viral transduction method. It is used to gene transfer method. Okay. So let's see the next question. Which type of viruses is commonly used in viral transduction for gene delivery? Options are retroviruses, bacteriophages, adenoviruses. So the correct answer is bacteriophages. They are generally used for the gene transfer method. Okay, let's see the next question. What is a potential advantage of using viral vectors for gene delivery? Options are low efficiency in delivering genes, high specificity for target cells, resistance to immune responses. So the correct answer is high specificity to target cells. Let's see the next category of questions that fill in the blank type of questions. Let's see the first one. Dash involves the use of tiny particles coated with DNA that are shoot into target cells using a gene gun. So, in a method, there we are going to use tiny particles. They are coated with DNA. They shoot into the target cells by using a gene gun. So that is nothing but biologistic method. It is also called as particle bombardment. That is the correct answer. Okay. Biologistic method or particle bombardment is the correct answer. Let's see the next question. Dash involves the use of tiny particles. So same question, this is also. So let's see the next question. In the chemical method of gene transfer, the use of dash enhances the entry of DNA into cells by forming complexes with DNA molecules. Okay, so the answer is liposome method or lipid method, lipids. Okay, so there the use of liposomes that enhances the entry of DNA into the cells. Okay, that is liposome method. Let's see the next question. Dash is a physical method of gene transfer where a fine needle is used to directly inject DNA into the nucleus of the cell. As I already told you, this method is nothing but micro injection method, by which the DNA is directly injected into the cell. Okay, so let's see the next category of questions. They are belonging to very short answer type of questions. You have to say the answer in shortcut manner, single line or a definition manner. The questions are going to be. Let's see the first question. Explain the particle behind electroporation as a method of gene transfer. So electroporation that involves applying of short high volt electric pulses to cells that creates temporary pores in the cell membranes. So that allows the DNA enter into the cell and it incorporated into the genome, which is a physical method. Electroporation is a physical gene transfer method. Okay, so let's see the next question. How does agrobacterium tumefaciens that participate in biological gene transfer? Yes, agrobacterium that transfers a part of Ti or Ri plasmids, that is called tDNA, into the plant cells. tDNA carries a number of genes. They are important for survival of agrobacterium and the bacterial infection in plants. This integrated DNA that carry foreign genes in the plant genome. Okay, in this way that is used in the genes transfer. Okay, let's see the next question. What role do liposomes play in the process of liposuction for gene transfer? So liposomes are the lipid vesicles that encapsulate the carry DNA, okay, into the cell. Okay, so in liposuction, these lipid DNA complexes fuse with the cell membranes that facilitates the entry of DNA into the target DNA. Okay, so encapsulated vesicles, they carry the DNA. They are nothing but liposomes. Okay, so let's see the next category of questions. They are matching type. You have to match the list 1 with list 2. Here I am going to match the list 1 with list 2. Electroporation method was discovered by the scientists Wang and Newman. Bialistic gene transfer, that is discovered by the scientist Sanford. Microinjection method was discovered by the scientist Marshall and Barber. Calcium phosphate method was discovered by the scientist Graham and van der Erb. So the correct option is fourth one. B.Sc. B.A. is the correct one. Let's see the next category of questions. They are belonging to true or false category. Let's see the questions one by one. Transduction is a method of gene transfer that utilizes electric shocks to create temporary pores in the cell membrane. So this is the wrong. Okay, so that means this is a true false statement. Okay, you have to write here false. Okay, let's see the next transduction is a gene transfer method that utilizes the viral bacteriophages. Okay, they are used to transfer the genes. Okay, so let's see the next question. Electroporation is a method of gene transfer that utilize electric shocks to create temporary pores in the cell membranes. Yes, this is true. Okay, this statement is true. Transduction is a chemical method of gene transfer that involves the use of lipids to deliver DNA to target cells. So again, this is also a wrong statement. Okay, so let's see the. Now I am going to share the questions with answers. You can take screenshots now. Okay. సో ఇవి స్టూడెంట్స్ క్వశ్చన్స్. సో ఈ ఈ వీడియో మీకు యూజ్ అవుతుందని నేను అనుకుంటున్నాను. యూజ్ అవుతుందనిపిస్తే గనక షేర్ చేయండి మీ ఫ్రెండ్స్‌తోని, లైక్ చేయండి, సబ్స్క్రైబ్ చేయండి, నా ఛానల్‌ని ఫాలో చేయండి. థ్యాంక్ యూ.